Hi, today I will do an experiment to show that photosynthesis does occur in plant cells. So to do this, we need to know how photosynthesis works. Photosynthesis is the reverse equation of cellular respiration, so it will take in light energy, and then it will also take in carbon dioxide and water, and then through a series of reactions, it produces oxygen gas and glucose, sugar, as products. So in our lab, we were able to see when gases are formed. This is an indication of photosynthesis in action because oxygen gas is a product of the reaction. To do that, we'll first suck out all the gases from a fresh plant cell, and then we'll give it some carbon dioxide and light energy. And then we'll see that as it does photosynthesis, it produces oxygen gas and it will start to float in a cup. So more in depth on how a plant leaf goes through photosynthesis, like what parts are there. If we draw a diagram of a simple leaf cell, you'll have multiple layers. The first layer is the cuticle, and under that you have a epidermal layer, and then under that you have the mesophyll. Mesophyll is where most of the photosynthesis takes place. You'll have a vein in the leaf. This is where the xylem and phloem are. Basically, that's where water and sugars are transported throughout the entire leaf. Then on the bottom you have the lower epidermal layer, and then you'll have the other cuticle. So this is a simple diagram of a plant cell. The cuticle on the very top is hydrophobic and water will not pass through it. So the cuticle causes plants to float on water normally, and then if you feel it and if you can see it, the shininess is because of the waxy property of the cuticle. And when we put water on it, water will not go into the cell. And because the cuticle is hydrophobic and it floats on water, it will not really help us in our experiment. So we need to make sure that we can get the plants to sink in the water. To do that, we will add a soap molecule. A soap molecule has two parts. The first part is hydrophobic, so it doesn't like water. The second part is hydrophilic, which likes water. So when we put a soap molecule onto the leaf, the hydrophobic region will attract to the hydrophobic cuticle, and then this will, and then so you see how there's a binding between the two. The hydrophilic region is still sticking out of the plant, and then this is what allows the soap molecules to basically grab a hold of the water and start to sink. And this is only like one molecule diagram, but remember that there will be a lot of soap molecules, and they will all attach to this cuticle, and basically it will form a sphere around the entire plant, and that's what causes the plants to sink. Okay, the second part, in order for plants to sink, is we need to get rid of all the gases already in it. So as I said, the mesophyll, this inner layer, the mesophyll is where the actual photosynthesis occurs. That means that it's going to have already gases just floating around because there's photosynthesis already taking place. So in order to get rid of all these gases and make sure that the plant leaf will actually sink, we're going to need to create a vacuum, and this vacuum will get rid of all of the gases within the mesophyll, and then the plant will sink. So to do that, first we will take a leaf. This is a leaf from our orange tree. We'll cut out holes in the leaf, and we'll do that using a hole puncher. So I'll cut out 20 discs. So I've cut out 20 leaf discs, which we will use in the experiment. Okay, so there they are. Let me find the small one. Okay. So there will be two groups in our experiment. The first is the control group, which is basically output these leaf discs in just plain water and shine a light source on it. And then the other one will have dissolved baking soda, which will provide carbon dioxide to the plants. And then I put that also in water and also under the same light source. And then we'll observe the difference between these two groups. So our procedure is first to prepare 200 milliliters of 0.2 molar bicarbonate solution. So bicarbonate, we get that in baking soda. So I did the calculations for it using stoichiometry. I've calculated that because sodium bicarbonate has a molar mass of around 83.89 grams per mole and has density of around 2,160 grams per liter. We can get that for 500 milliliters of water, I'll need 0.1 moles of sodium bicarbonate, and that amounts to around 8.39 grams, which is around 3.88 milliliters. So using those calculations, we measured out 3.88 milliliters of baking soda. And now I will prepare the molar solution. So I said 500 milliliters of liquid will be used. So this is our 500 milliliter beaker. First we pour in the 3.88 milliliters of sodium bicarbonate. Okay. And then we will fill this beaker with water up to the 500 milliliter mark. Okay, so that's approximately 500 milliliters of water and around 0.1 moles of the solute. And then we'll stir this to get more spread out evenly. Use a spoon to do it. Okay, so this is our 0.2 molar solution of sodium bicarbonate, which will give us the source of carbon dioxide that the plants will use in their photosynthesis. Now, our cups, we're gonna put 200 milliliters of this solution in, only one of the cups, and that will be our group with carbon dioxide. So we have a small beaker. This measures 100 milliliters, and we will measure 2 of these beakers, and then we'll pour it into a cup. Okay, so So that's 200 milliliters of the bicarbonate solution. And then we'll label this cup with the words with CO2. And then this is a cup with 200 milliliters of just pure water. This is without carbon dioxide. This way we can differentiate which group is which. Okay, next we have to put in the diluted soap in order to make sure the leaves will actually float and sink. So we only need around like 1 drop of this solution. This is diluted soap. And then we drop in 1 drop. And then we start. Okay, so both of these cups now have a bit of diluted soap, which should help the plants sink or float. And then next step is to actually draw the gases out of the plant leaves. So this is a syringe, right? It has a plunger. How we're going to draw out the gases is by creating a vacuum, and then you can create a vacuum by sticking your thumb on top of the syringe without a needle, and then you pull the plunger back like that, and that helps create a vacuum. So to do that, let's fill this syringe with water. And then we'll put in 10 leaves. And then we put the plunger back in. So you can see the leaf discs, most of them are floating. And that means that there's still gases within their mesophyll. And then to draw the gases out, we will create the vacuum. So put my thumb on the tip and then draw the plunger back. Do it a couple of times until they all sink. As you can see, some of them are sinking. There's still 2 floating. Okay, so now the leaves have sunk. This means that the vacuum has drawn out the gases within the plant leaf. Now we'll put these 10 leaves into the first group, which we'll do is without carbon dioxide. So you can see they're sinking. Okay, and then we'll repeat that step with the syringe for the other one. Also, it might be better because now there's a delay. We should cover the first cup so that no light enters and then no photosynthesis will occur. Okay, so this is the second batch of 10 leaves. Now they are all sinking. Alright, and then now we will put all of them in the second cup. Okay, and then now we'll bring both cups outside where they'll do photosynthesis under natural light. Okay, so we have our cup with carbon dioxide from baking soda and our cup without carbon dioxide. And now we go outside. Okay, so as you can see, most of the leaf discs in the cup with carbon dioxide from the baking soda has risen. It's that clump over there. And then the ones in the cup without the carbon dioxide, they're still on the bottom. So it's not particularly sunny today outside, but there's still enough light to get the photosynthesis running. And then because of this, you can see that the products for photosynthesis are met on the cup on the right, because it has light energy, and it also has water, and it also has carbon dioxide. Oh, you can see the last one's floating. And then as the products, there should be sugar within the cells now, and there's also oxygen being released. And then the oxygen being released is what propels these leaf discs to move up and go to the top of the water.